All right, picking up from where we left off yesterday, we talked about the structure of the chloroplast with the double membrane thylakoid, so the network of membranes, the thylakoid and the grana and the lamellae. Then we talked about different pigments inside the chloroplast, generally known as chlorophylls, but there are a number of them. So you've got chlorophyll A, chlorophyll B, carotenoids, and phyophytin. The absorption spectrum, which tests and indicates the wavelengths of light, visible light, that are most absorbed by the photosynthetic pigments, and they are in the blue and red region of the spectrum. And the action spectrum, so those are the wavelengths of light at which the highest photosynthetic activity is recorded, and not surprisingly, and as expected, in fact, that is also in the blue and red region, because you would expect photosynthesis to occur in the with the light that is absorbed. Then we talked about chromatography, and this is a technique used to separate different pigments or different solutes in a solution. In case of the plant leaf, to figure out which ones are present in a particular leaf, we grind the leaf with the right chemical, then take the filtrate, and then use that filtrate on a chromatography paper or silica gel, and then perform the experiment, and the solvent will travel up the paper to a certain height, and along with the solvent, the solutes will travel also up to a certain height depending on their mass and the structure. Then we calculate what's called an RF value, which is the distance moved by the solute, so from the baseline up till the end. The distance traveled by the solute, so that is from the baseline up till whichever point a particular solute traveled, divided by the distance moved by the solvent, so that's from the baseline till the end. And RF value will be anywhere between 0 and 1. It can't be greater than 1, and obviously can't be less than 0. And to identify which solutes you have, or in this case, which pigments you have, we compare them to known RF values for a particular solvent, because how far they move up the paper or the gel depends on the solvent that is used. Now today we will talk about photosynthesis, and what we're going to start off with is, I believe I mentioned this yesterday, that photosynthesis occurs in two different stages: the light dependent stage and the light independent stage. So photosynthesis and you have the light dependent stage happening first. So yes, light is important for photosynthesis, not for the entire process, but for the first step. And it is the products of the first step or the first stage that are actually used in the next stage, which does not require light, the light independent stage, to produce glucose, right? And so, right, if you don't have light, you can't proceed with photosynthesis. The light dependent stage, right, obviously involves the chloroplasts. And within the chloroplast, the pigments are arranged into two complexes. These are called photosystems 1 or PS1 and photosystem 2. Right, see that the 1 and 2 is being written with Roman numerals. Now, the fact that we have 2 of these guys means that they are working slightly different. Okay, and what's happening is that in either of these, in the light dependent stage, what happens is that light is taken up by the pigments in the photosystem and are used, right? So light is absorbed by pigments in the photosystem. Right? And used to, I'll add these in bullet points. In fact, and used to split water so that is a photochemical reaction. That is one word, by the way. Write it closer. Photo meaning light and chemical meaning chemical. So it's a chemical reaction that involves light. Right? So it's water is split. That's a photochemical reaction. And what that does, it provides hydrogen ions to reduce CO2 to carbohydrates, to form carbohydrates. Right? But that happens in second stage, the light dependent stage. So the hydrogen ions are produced in the light dependent stage. They're used to reduce the carbon dioxide to carbohydrates in the light independent stage. So, right, they're taken to the next one. Also, right, electrons are used to keep the photosystem running. So we'll talk about that detail when we talk about each in a bit more detail. Also, ATP is produced. ATP is produced, right, and that provides energy to build the carbohydrates. But as we've seen, the photosynthetic process requires energies, right? So it has an enthalpy change, as a positive enthalpy change. So it's taking up energy. Now, this ATP that is produced is minimal, right? So it's only produced in like as much in the process of photosynthesis that would be utilized to build the carbohydrates. So this is not the plant's primary source of energy, right? The primary source of energy for plants, as we've talked about, is respiration. Any questions?
No, sir.
Okay. Now, right, all of this happens in two different processes. There are two different processes occurring in the light dependent stage, and right, there is no sequence between them to say that one happens first and then the other happens next. And there's no preference between them that under certain conditions, one would be favored over the other. Right, so I'm saying that before going to explanation, that they both just happen and they happen simultaneously. Okay, and the first one is called cyclic photophosphorylation. Cyclic, we know what cyclic means, right? It's a process that is, right, keeps going round and round, right? Keeps repeating. It starts and then feeds back into itself. Not like a positive feedback loop, it just, replenishes at the end, like we did the Krebs cycle. And there's something we'll come across today, which is known as the Calvin cycle, right? So these are cyclical processes, and right, they replenish themselves at the end. Photo again means light, and phosphorylation means phosphorylation happening. In this case, it refers to the production of ATP, right? A term that we are familiar with. We've come across phosphorylation, oxidative phosphorylation, when we talk about respiration. Okay. So what happens in cyclic phosphorylation? Let's talk about this one first, and then we'll talk about the other process. So what happens is light falls on chlorophyll. Right, if I just change colors for a bit. So light is absorbed by chlorophyll. Right, what that does is, right, so light is made up of packets of energy called photons. Photons. Right, and these obviously have energy. And that energy is transferred to the electrons of chlorophyll. Now, when the electrons gain energy, they become excited, right? High energy, so excitement, we can understand that. And if they gain enough energy, they actually leave the chlorophyll molecule. Okay, what do they do when they leave? Well, they are picked up by a series of electron carriers, right, or electron acceptors. And right, they move along that series, and as a result, right, the energy that the electron have, right, the raised energy, is used to convert ADP and inorganic phosphate to ATP, right? That's the photophosphorylation, right? So this is photophosphorylation. Phosphorylation. Right, so ATP is produced, but the electron's not done. So the electron right? It travels along the series, and the electron is returned to the chlorophyll molecule. And that's what makes this process cyclical. Any questions?
None, sir.
Okay, so simple enough, right? And especially,
Yeah.
having gone through respiration, this might seem like, oh, that's it,
Yeah.
right? That's
Yeah.
all. That's nothing. Okay.
Yeah, it's like basic sum up, I think.
Yeah. Oh, by the way, right? We talked about the two photosystems, right? In cyclical process, this cyclic photophosphorylation, it is only photosystem 1, PS1, is involved. Only PS1. And by the way, right, in the book, if you read it, it's written something like this. I would suggest when you're writing it out, you write it either with a hyphen like I have, or you write it with enough space to make it clear that the 1 is not part of the PS, just to avoid confusion of any possible sort, right?
Okay, sir.
I would say that it could be confused with the pressure per square inch, PSI. That's something that's used in physics, but that's all written in lowercase, so you really can't confuse it with that. But still, right? It's
Yeah.
better. I mean, there's no standard way of writing this. So either give a space, or better yet, put a hyphen that just makes it clear.
Okay.
So that is, right, cyclic or cyclical photophosphorylation. Then we have the other process occurring, which, if you would guess how these things are named, if one is cyclical, then what would the other one be? Any ideas?
No, sir.
Oh, come on. One's cyclic, so the other is non-cyclic.
Oh,
Sometimes scientists aren't very creative with their names, and in the grand scheme of things, that's actually a good thing, right? It's very plain and simple. uh, just an example on, like, uh, you've obviously, about uh, the Arctic and the Antarctic, right?
yeah.
I didn't recall which language those words are taken from. They're not English, right?
Yeah.
Latin, perhaps, I'm guessing. You know what Arctic actually means?
Arctic?
Arctic.
Something with Earth's region or ocean,
Oh, no,
something
no,
like
no.
that.
The Arctic, right? Where you at the poles, where you've got lots of ice, right? And
Yeah.
glaciers and stuff, icebergs.
Mm.
The Arctic, the word Arctic means bears, right?
Oh,
So at the pole, at the Arctic, one of the poles, right? You have bears, and so whoever went there, whoever discovered the Arctic, he was like, "Oh, bears! Let's call it bears," right? They went to the other pole, and they called it Antarctic. Now, what do you think that means?
something similar?
No bears,
Wow.
because there are no bears in the Antarctic,
Yeah.
right? So even though it sounds fancy because it's not English, well, English is a combination of many other languages over, long periods of time, etc., etc. But still, right? It actually translates to something very simple: Arctic and Antarctic, bears and no bears. So cyclic photophosphorylation and non-cyclic photophosphorylation.
Yeah.
Photophosphorylation. Will you have to keep track of the spelling? You can easily get lost halfway through and be confused in what you're writing.
Yeah.
Just look at that thing, photophosphorylation. Okay. Anyway, so as the name suggests, right, it's not a cyclic process, right? And this one involves both photosystem 1 and photosystem 2. And the story begins with photosystem 2, right? So, I mean, technically it's all happening together, but to understand the flow of events, we'll start with photosystem 2. So what happens, just like in cyclic photophosphorylation, light is absorbed by chlorophyll in photosystem 2, right? So over here, light is absorbed by chlorophyll, right, in photosystem 1. So and right, the electron gains energy and it leaves the chlorophyll. Again, right, the term chlorophyll is being used as a general term for all the pigments that are in photosystem 1. Photosystem 2 or 1. So, right, the electron leaves the chlorophyll. Okay, sure. Then it is picked up by an electron acceptor, right, and moves through the electron transport chain. So picked up by electron acceptor, and moves across the electron transport chain. And as with the cyclic process over here, ADP, and inorganic phosphate are combined to form ATP. So that's the phosphorylation part, but that doesn't have stop here. What happens to the electron in this particular process? The electron, right, is taken up by photosystem 1. Right? Again, light energy, excited electron, high energy electron, right? Released or leaves. Now what happens is, right, that electron is picked up by NADP. It is taken by now does NADP look and sound familiar?
Yeah.
Right, we've come across NAD and FAD. So nicotine amide adenine dinucleotide and flavonamide dinucleotide, right, in respiration. Well, this is of the same family, right? It's NADP. So nicotine, amide, actually one thing. Adenine dinucleotide. And what do you think the P is?
I'm not sure.
Oh, come on, take a guess.
Phosphate?
Phosphate.
Okay.
All right, so that's what NADP is. Right, now we know that, right, NADP is not only an electron carrier but a hydrogen acceptor. Right, so it's an electron carrier and hydrogen acceptor. And so far it's only picked up the electron. Right, now it's not done yet. Right, And again, this is not like, oh, this happens first and then the other thing happens first. It's all happening together. It's very fine tuned, right? Now, right, what's happened is the electron started off from photosystem 2, went across the electron transport chain, ATP was produced, was picked up by photosystem 1, released again due to light energy, taken up by NADP. Now, NADP to become reduced, right, still needs the hydrogen. The hydrogen is missing. Secondly, right, photosystem 2 lost an electron, right? So an electron was lost from one of the pigment molecules. And that's not something we want, right? Because we need to replenish that, right? Because what's actually happening is photosystem 1, the electron that it lost wasn't like, lost only after it received it from photosystem 1. Because that would mean that it got an additional electron which it released. No, as photosystem 2 release the electron by gaining light, light is not falling in turns on photosystem 1 and photosystem 2. Light is falling on the leaf, it is falling on the chloroplast, it is reaching the chlorophyll in both photosystems, and so simultaneously electrons are being released by both photosystem 2 and photosystem 1, right? And so this process is happening at the same time. The electron from released from photosystem 1 is taken up by NADP. Okay, right now it's one electron short. Where does it get that electron back from? It gets it from the electron that was released by photosystem 2, as we just saw, right? Released from photosystem 2, travels electron transport chain and taken up by photosystem 1. So gains back the electron, becomes stable again, becomes complete again. Okay, but then what the question is, well, what about the photosystem 2? Where does it get its electron back from? And that's where we talk about photolysis, right? So we talked about the splitting of water, the photochemical reaction. That's what's happening in the non-cyclic photophosphorylation. So, right, light energy. Are you following along?
Yes, sir.
Is used to split the water molecule. Right, water is important for what you call it, photosynthesis, right? It's carbon dioxide plus water. That's what the water is doing over there, right? To split a water molecule. And this is called photolysis, right? Photo meaning light, and lysis meaning splitting or breaking up. So light is being used to split or break up the water molecule. And how does it do that? Right? So, right, you've got H2O and light, and it splits it into the hydrogen ion and the OH ion. Okay, now changing colors again. This hydrogen ion, right, is taken up by, can you tell me? Where does the hydrogen go? We just talked about it.
Um,
Just talked about it. Hmm? No? Kind of lost?
yeah.
Well, the NADP. Right? This guy over here. NADP, nicotinamide adenine dinucleotide phosphate. So it got the electron from photosystem 1. It still needs a hydrogen. Right? So taken up by NADP, right, to become reduced NADP. Right, so what's happening is, in case of NADP, you've got NADP plus electron plus proton, right? So basically that's hydrogen, right? Because hydrogen, the hydrogen atom is just an electron and a proton,
Yeah.
right? To become reduced NAD.
Okay.
Okay. Now this electron, remember, this one's coming from photosystem 2, sorry, 1, right? And this one is coming from photolysis.
Okay.
Now, what about the OH? Right? So all these OH ions that are created are processed, right? They lose an electron, and this electron goes to photosystem 2. So when we started off, photosystem 2 lost an electron. Yes?
Yeah.
Photosystem 2 lost an electron when we started off with the non-cyclic photophosphorylation. It's going to gain that electron back from the OH ion that we got from water. Okay?
Okay.
And what happens overall to the OH ion? Right? So let me just write that down. Right? So 4 of these guys are used, right? They each lose an electron, so 4 electrons are lost. And what you get is a molecule of oxygen and 2 water molecules back. And that is the end of non-cyclic photophosphorylation and the end of the light dependent stage. Now, right, does this seem like it's a bit confusing?
It's okay, I guess.
Okay, very quick recap. Cyclic photophosphorylation involves only photosystem 1. Light is absorbed by the chlorophyll in photosystem 1, right? Photons have energy that's transferred to electrons, which gain energy, they become excited, and when they become excited enough, they are released from the chlorophyll molecule, right? Picked up by a series of electron carriers, where the energy of the electron is used to make ATP, and the electron is eventually returned back to the chlorophyll molecule, so that makes it a cyclical process. Non-cyclical photophosphorylation has a few things going on, right? It involves both photosystem 1 and 2. To understand it stepwise, although all of this is happening simultaneously. Light is absorbed by chlorophyll in photosystem 2. Again, electron gains energy, is released by the chlorophyll molecule, picked up by electron acceptors, moves along the electron transport chain. Its energy, just like right in the other process, is used to make ATP. That electron is taken up by photosystem 1. Right now, independently, right, this electron that's taken up by photosystem 1 is actually being used to replace the electron that was lost. Why? Because at the same time as photosystem 2, photosystem 1, sorry, also absorbed light. And similarly, right, it got its electrons in an excited state and released them, right? And that one was taken up by NADP, which is nicotinamide adenine dinucleotide phosphate, right? So the electron that's being taken up by photosystem 1 is actually replacing the one that itself lost. Now, we have two things that are incomplete. NADP has taken up an electron, but still requires hydrogen because it is a hydrogen acceptor, right? So it's missing a hydrogen. And photosystem 2 is also missing the electron that it lost, right? So it needs stability, it needs to replenish it. Remember, this is not a cyclic process, right? The electron is not going to come back from photosystem 1 because it's taken up by NADP, and that's it. So where does photosystem 2 get its electron from? Where does NADP get its hydrogen hydrogen from? And that comes from the water molecule, right? So light energy is used to split the water molecule into a proton and the OH ion. The proton is taken up by NADP to become reduced NADP, so that's one thing that's settled. The OH molecules are, right, they are combined through a chemical process. So they each lose, so four of them, to write out a balanced equation, four of the OH ions each lose an electron, so 4 electrons are lost. And then they are combined to form a molecule of oxygen and water, right? So, right, this is the oxygen that is either, generally we say is, oxygen is produced during photosynthesis. This is where that oxygen is coming from. It's actually coming from the splitting of water, right? And some water is replenished, so to speak, right? Not the whole thing, but some of it, right? So 4 are used to we get back, right? So 50% is reattained, and this is in the form, right? It could be given out as water vapor, or it could be utilized by the plant cells. Similarly, oxygen could be released as an excretory product, or it could be taken up by the plant cells for respiration. Okay,
Okay.
so that is the light dependent stage, right? And after this, the next time we will delve into the light independent stage, right? And after that, talk about limiting factors, and then we'll be done with the chapter of photosynthesis. Any questions?
No, I'll try to ask questions of this tomorrow.
You mean next class? We don't have one tomorrow.
Yes.
Okay. All right, sure, yeah, but
And
definitely go read the book as well, right? So I'll make the recording available. You can refer back to that, and right, you are making notes definitely, and then
yeah.
read the book.
Yes, sir.
Okay, and again, right, to understand all of these things, as I mentioned earlier, right, make your own notes, and especially for these processes, write them down, or if they're like drawn in a series, then draw them out yourself. Helps with the understanding and the memorization.
Yeah.
All right.
Okay.
Okay. All right, then thank you, and I'll see you next time, inshallah.
Yeah. Gracias.
Allah.